Welcome to the channel. Today, we're looking at the math behind how doctors calculate medical risk. In breast cancer prediction, there is a major variable, rare, high-risk genetic mutations, specifically in genes like BRCA1 and BRCA2. The second major variable is mammographic density. This is a physical measurement, the ratio of dense fibroglandular tissue to fatty tissue in the breast. For years, the clinical assumption was additive. If a patient's mutation and highly dense breast tissue, those two factors would compound, pushing the patient's overall danger level higher. But predictive models lacked hard data on how these specific elements interact. We haven't known what actually happens inside the risk equation when a patient presents with both a mutated susceptibility gene and highly dense tissue. Precision medicine requires precise math. To build algorithms that accurately protect patients, oncologists need to know if these genetic variants physically cause denser breasts, or if these two variables operate entirely independently of each other. A 2026 pooled analysis published in the Lancet tackled this exact problem. The Breast Cancer Association Consortium gathered data from a massive cohort of over 24,000 cases and controls. The researchers lined up the women's precise genomic sequencing data alongside automated, digitally mapped measurements of their mammograms. The data showed absolutely zero association between carrying a pathogenic gene variant and having denser breast tissue. Because a mutation in a gene like BRCA1 does not physically produce a denser breast, risk algorithms have to treat genetics and density as completely separate, non-overlapping inputs. The next step was to see what happens when both of these independent variables are fed into a predictive risk model at the exact same time. Intrigued, the mathematical interaction between genetic burden and breast density is actually negative. For the patient, this means the relative danger added by high breast density is significantly weaker in women who carry high-risk genetic mutations than those who don't. Possessing a high-risk gene dilutes the predictive threat level of that patient's physical breast density. To verify this observational data, researchers applied a rigorous statistical method called Mendelian randomization, which uses genetic variation to isolate cause and effect. The math points to a specific biological mechanism. Mutations in genes like BRCA1 strongly predispose women to a very specific tumor subtype, known as triple-negative breast cancer. Triple-negative breast cancer behaves differently than other tumors. The data shows it is far less influenced by physical mammographic density than estrogen receptor-positive tumors are. The genetic mutation dictates the specific type of cancer a patient is likely to develop, effectively bypassing the physical density risk factor in the process. These findings validate existing risk algorithms, like BOADICEA, in their approach to treating genetics and density as separate, independent factors, but it provides an important clinical correction. Doctors will need to specifically adapt their risk calculations for genetic carriers who also have dense breasts to prevent overestimating their actual danger. The math of precision medicine has become more exact. By understanding exactly how these variables interact, oncologists can profile their highest-risk patients with much greater accuracy. Thank you for watching. Please like and subscribe to this channel and press the bell icon for more updates.